All right. So we are finishing up our mitosis notes, our mitosis foldable. And I was hoping to start binary fission, but with the lockdown drill, we will see how far we get. Does anybody have any questions before we get started? Literally anything? All right. Where were we? Did we finish up metaphase? Yes. Okay. So anaphase. Make this actually. I'll start with her. No. Ah. All right. So anaphase, our circle is going to be a little bit elongated, kind of ovalish. I believe our centrioles were green. So we're going to have those still at either end of the cell. The little asters. And it is anaphase. I'm not sure if I told you yesterday, but I have like little mnemonics for metaphase and anaphase. It's like metaphase meet in the middle, anaphase apart, because I like alliteration. So in anaphase, our chromosomes are separated and pulled apart. Green. So each chromosome is split into two sister the sister chromatids are split into individual chromosomes. And we have little connectors like that. That is anaphase. Again, anaphase is, and telophase are probably the most familiar looking phases of mitosis. They're pretty much the same. They aren't changed like prometaphase and prophase are, but yeah. So we still have our centrioles, our centromeres, connected via those microtubules and pulled to either end of the cell. So, cool here. All right. Ah. This is our shortest phase. Meaning, if we were to take a screenshot or freeze a fast-growing tissue in any moment of time, the amount of cells that we find in anaphase is going to be the least. So I have a little correction for you. Earlier, I think it was Monday or Tuesday, I said that the sister chromatids are held together by cohesion. Read that wrong. It is a protein called cohesin. So, cohesin proteins are what normally holds the sister chromatids together in a duplicated chromosome. So, in this step, those cohesin proteins are cleaved. So now sister chromatids part. And they are now independent chromosomes. So our sister chromatids have been pulled apart. And we now have two independent chromosomes. And those chromosomes are moving towards opposite ends of the cell as those kinetochore microtubules are shortening. So chromosomes move to opposite ends of the cell. As kinetochore microtubules shorten. Never know which side of the thing to stand on. So, the act of pulling those chromosomes from one end to the other isn't necessarily a pulling motion, but it is the microtubule shortening. So, as monomers are taken off of that giant microtubule, it shortens, and as it shortens, it brings that chromosome closer to either end. The cell elongates because the non-kinetic microtubules lengthen. So these microtubules are shortening and these microtubules are getting longer. So the cell elongates as non-kinetochore microtubules elongate. And then at the end of the phase, either pole has equivalent and complete sets of chromosomes. At the end of the phase, each pole has equivalent and complete collections of chromosomes. My grammar's off here and I can't figure out how. Ah. So, try to make this easier to hold. Goodness gracious. It's not functioning. Okay, finally. We have telophase and cytokinesis that we have kind of lumped together. So we're gonna, because so the cell cycle isn't necessarily one of those things where it happens like this stage happens and then this stage happens and then this stage happens and then this stage happens. There's some overlap. One will start as one stops. It just kind of flows from one phase to the other. So telophase and cytokinesis are often lumped together. So what we're going to draw, our elongated cell has started to pinch in. This pinch is actually called a cleavage furrow. Because the process of one cell becoming two, cytokinesis, is also called cell cleavage because they are cleaved down the middle. We still have our asters Our chromosomes, we only have one little arm in each of them, but they're also starting to become a little wiggly. You could even draw them fuzzy if you wanted to. And we have our nuclear envelope starting to reform. All right. Cool. Okay. So we're going to start with just telophase. So I have like two separate headings in the flat. So in telophase, we now have our nuclear envelopes reforming from portions of the old cell's endomembrane system. New nuclei forming from parts of the old cell's endomembrane system. So parts of the nucleus from the first cell that were dissolved, parts of old Golgi apparatus, parts of old vesicles, parts of old endoplasmic reticulum will come together to make sure we get a fully functioning nucleus. Our nucleoli are going to start to reappear. Because our chromosomes are becoming less condensed. Our chromosomes are becoming less condensed. Oh my goodness, my handwriting. Whoops. All right. So I gotta be neater. Our microtubules, whatever's left from the microtubules that were pulling on the chromosomes, are depolymerized, meaning broken up into pieces. So remaining microtubules are depolymerized. This means mitosis is complete. This means mitosis is complete. Remember, mitosis is just the division of the nuclei, and at this point in time, our nucleus is completely separated into two. Getting it back together. All right. That's telophase. Now for cytokinesis. This is underway in late. Cytokinesis is the division of the cytoplasm, and it is underway by late telophase. So, division of cytoplasm. This is underway by late telophase. So even if mitosis is not complete, cytokinesis can at least begin. And it's a little bit different in plant cells and animal cells. We have been working with animal cells. All the cells that we drew in this diagram are animal cells. We know because we do not have a cell wall, and we have the little centrioles. Remember, plant cells don't have centrioles. So in animal cells, we get the formation of a cleavage furrow. And that will pinch the cell into two. Let me triple check something really quickly. Make sure that I'm not going to tell you wrong. Perfect. Yeah. So in animal cells, it's a little different, or not animal cells, in plant cells, instead of the cell pinching into two, a cell plate will form between the two new cells. A cell plate forms, dividing the cell in two. So there's not really a pinching process with a plant cell. What will happen is a cell wall will form between the two, and the cell membrane or the cell plate will form between the two. Cell membranes will grow around that plate on either side, and a cell wall will form around that plate on either side. So that's how that works. Anyone have any questions? That is mitosis. So we don't have a heck of a lot of time, but I do want to at least start what I had planned for today, which was talking about binary fission and the evolution of mitosis. So, if you would like, what you could do is if you take approximately half of your foldable, it should fit. It should fit if it's like folded in half on a single page in a notebook. You might have to do it horizontally instead of vertically. We can glue that in there. That way, when you need it, you can just whoosh, open it right up. I have glue sticks for that too. No, you don't at all. You can keep it wherever you want. But if you just had a notebook instead of a folder, you can keep it in there. Does anybody want glue stick? Mm-hmm. Mm-hmm. All right. So, binary fission and the evolution of mitosis. We'll probably just get to binary fission today, which is a-okay. Sorry, my sock's falling around the back of my foot. All right, so everything we've talked about with mitosis is the process of cell division in eukaryotic cells. But prokaryotic cells work a little bit differently. I'll give you time to get that down. So binary fission is the process of asexual reproduction in prokaryotes, where essentially cells grow double their size, divide, and form two cells. So binary fission is the process of asexual reproduction in prokaryotes where cells grow to roughly double their size and divide in two. We don't have the act of mitosis happening. We don't have these distinct phases. In fact, we don't actually know a heck of a lot about binary fission. We know the basics, we know how it happens, but it is actually super, super quick. Like, it's a fast process, which I think has something to do with why we haven't really nailed it down yet. It's also really simple compared to mitosis. And usually in biology, when things seem simple, it's kind of hard to nail down the actual complicated reasons behind it. So binary fission is the process of asexual reproduction in prokaryotes where cells grow to double their size. Let's see. I don't know if we really talked about the difference between asexual and sexual reproduction in this class yet. In biology, the prefix A just means without. So asexual reproduction is reproduction without sex. We are talking specifically about single-celled organisms when we're talking about prokaryotes, but other organisms can undergo asexual reproduction as well. Plants are a really big example of this. For a while there, my TikTok feed was just a bunch of people stealing plant clippings and propagating them at home. So the act of plant propagation is actually asexual reproduction. If you take a leaf off a plant in a grocery store or nursery, bring it home and plant it, you're not adding any DNA to that new plant. So that whole new plant that you're growing at home has the exact same genetic information as the plant that you stole it from, which is intuitive when you think about it, but actually kind of really cool. So binary fission is reproduction or cell division in prokaryotes. Bacteria has most of its DNA in a single circular chromosome, which is another reason why it's a lot simpler to divide a single cell because they only have one chromosome and oftentimes it is circular. Bacteria has most of its DNA in a single circular chromosome. The reason they say most right here is because bacteria have a way to share genetic information without reproducing. So one bacteria can share what they call a prion, which is just a tiny circle of DNA, to another bacteria. And that's not necessarily stuff that that bacteria needs to live, but can be cool stuff like antibiotic resistance. So that's why they say most. So they have one big chromosome, but sometimes they have little baby chromosomes or little baby circles of DNA that don't really have to do with reproduction. Really ate into our time today. Sometimes cell division is triggered by replication, and this is all prokaryotic division. So binary fission can be triggered ooh, that's not a letter. By DNA replication. Meaning, whereas in mitosis, right, we have that G1 phase, and once we get past that G1 phase, we signal our DNA to divide, and then we have the signal to start mitosis. But it can actually, or prokaryotes can actually start the process by just like, if the DNA starts replicating, okay, then we're splitting kind of thing. Let's see. This happens at a certain place on the chromosome, or replication starts at a certain place on the chromosome. Replication starts at a certain place on a chromosome. Called something really super creative, the origin of replication. Yeah. So the circle has a specific spot where that DNA replication starts, and that one special spot is called the origin of replication. It is the same. So origin of replication on this chromosome is going to be the exact same spot of the origin of replication of the chromosome next to it. I think that's where I want to call it for today. Is all right. We will finish this up tomorrow and hopefully start what I had planned for tomorrow. We'll just keep reorganizing. What I have for the next 2 weeks. I think we're just going to end this unit like we've ended all the other units. So nothing gigantic, nothing big. And then we'll kind of lean harder into AP prep next semester. So we've got binary fission and evolution. We are going to talk about cell cycle regulation and control. We're going to talk about what happens when we lose cell cycle control. We will review all this, do the progress questions, review the progress questions, and that will take us to the end of the semester. Next week's last week. So we'll get all that in. We'll probably do Monday, maybe Tuesday if we have to keep pushing stuff. Will be new content, review, progress questions, review the progress questions. So nothing too crazy. The next semester we'll probably do like actual exams based on AP questions, just to make sure that y'all are ready for that big test.